Hey guys, welcome back to Now I Know. Today we are talking about Maxam Gilbert method of DNA sequencing. Now first thing first, what is DNA sequencing? Now as the term itself says, DNA sequencing is a method by which I can sequence the DNA or know the sequence of a DNA. That means it's a process. If this is a DNA piece or DNA strand that I have, I'll be able to read the nucleotides that are present in this stretch of DNA, a process by which I can determine the order of all nucleotides. That is called DNA sequencing. So any method that would help me to determine the sequence or determine the order of nucleotide is called DNA sequencing. Today we are learning or starting with Maxam Gilbert method. Now Maxam Gilbert method was given in 1977 by Allan Maxam and Walter Gilbert, and it utilizes chemical to read the sequence. Okay, it will use the chemical approach to read the sequence. So it is also known as chemical method or chemical cleavage or chemical degradation method. Why cleavage and degradation? We will understand once we see the procedure. So in this video, we will see what is the mechanism of Maxam Gilbert method, how these chemicals help us to read the sequence, and in the end, we'll take an example and we will see actually how to read the sequence. We'll learn from the result that we obtain how can we read or determine the sequence. So let's begin with today's video. Now, before we start with the procedure, there are 2 points that we should understand. The first point is when we have the DNA, when we are going to read this DNA, we need to have certain tag or certain labeling which would tell us that whatever sequence that we are reading is from this particular tag. Okay, suppose I'm reading ATGC, that nucleotide is away from this particular tag. It should specify the direction in what we are reading the nucleotides. Okay, so tagging would help us to determine the specific location or position of that nucleotide in one particular direction. And when it comes to DNA, the best tagging method would be tagging the DNA with radioactive phosphate. Second point, this DNA that we are going to work with is in the double strand form. So reading double strand DNA is going to be a problem. So we need to have the sample in single stranded form. Of course, if we know sequence of one strand, the other is going to be complementary. So we need to keep these 2 points in mind. Now let's look at the procedure. Now largely we can divide Maxam Gilbert method in 2 parts. This is the way I remember. One is preparation of DNA, a single stranded labeled DNA, which is ready to be processed by chemicals. And second part should be when you take this single stranded labeled DNA and treat it with chemicals. That would give us the sequence. Alright, so first we will see how we can prepare the DNA sample. Alright, so as I said, this is the sample DNA. Of course, it is going to be in double stranded form. First thing that we need to do is label it so that we know that we are going to read it in this particular direction only. And for that, we are going to label this DNA with radioactive phosphate. Now, to add a radioactive phosphate, first we need to remove the existing normal phosphate from 5' end, both the 5' end. So to remove the phosphate, the enzyme that we use is phosphatase. I hope we all know this. So we will use alkaline phosphatase that removes the phosphate from 5' end. So it will dephosphorylate at both the 5' end. Now we have the DNA without the 5' end phosphate. Now we'll add our radioactive phosphate. And to add phosphate, we need kinase enzyme, that also we are clear about. So we will use nucleotide kinase, that means it adds phosphate or rephosphorylate with radioactive phosphate at both the 5' end. So now our sample is ready, and what modification we did over here, we radioactively labeled it. At 5' end there is a radioactive phosphate. So now we know that whatever sequence we are going to read is from this labeled 5' end. So there will be no confusion. Now the first point is done, but still it is in double-stranded form. As I said, we need a single strand form to read it, and the easiest way to denature the double-stranded DNA to single-stranded DNA is by applying high temperature. So we denature this double-stranded DNA to get single-stranded DNA sample. Now at this particular stage, we have denatured our double-stranded DNA and they are in single-stranded form. Here what we need to do is separate each single strand DNA and purify them for the next step for reading the DNA sequence. So what we need to do is, now this single stranded DNA, run it on gel, electrophoresis, subject it to electrophoresis. All the single strands will be separated. This is a schematic representation. Once these single stranded DNA are separated on the gel, go for extraction of this DNA. Now here, if you guys don't know, you can extract the DNA from this gel, right? Wherever you locate your single stranded DNA, you can chop it off this piece of gel, and there's a whole lot of procedure that you need to follow in order to purify the single stranded DNA. Now at the end of this, we are going to have our single stranded DNA ready for processing. Okay, what I did was, first label the DNA with radioactive phosphate. Then from double-stranded we got single-stranded, and to extract the single-stranded DNA in pure form, we went for electrophoresis and gel extraction. At the end of that, we are going to have a single-stranded DNA sample ready for processing. Now what we will do, this was the first part, as I said, preparation of a sample. Second part is, we need to treat it with chemical. Now there are 4 nucleotides that we are looking for, right? And we need to process the sample for each nucleotide separately. That's how only we will know where that nucleotide is present. So now we will divide this sample in 4 aliquots, and for each nucleotide, to detect each nucleotide, we need 4 different chemicals. That means each tube will be treated with different chemicals. So now let's see how to do that. So the basic mechanism behind this method is to use chemicals that can remove the particular base. Okay, and that's why it is called chemical degradation or chemical cleavage method. And how that works is, as I said, we need 4 different chemicals for 4 bases because they are going to be specific for that particular base. Say for example, dimethyl sulfate can modify guanine. And when I say modify, we are not going in detail to see how they modify. Just remember modify. Say for example, dimethyl sulfate can add methyl group to guanine, and that's how it is going to modify it. It methylates guanine. So dimethyl sulfate can modify guanine. Similarly, formic acid has the ability to modify adenine and guanine. Okay, hydrazine can modify cytosine and thiamine. When we add hydrazine plus sodium chloride, it will modify only cytosine, because sodium chloride would inhibit the effect of hydrazine on thiamine. So if we add combination of these two, hydrazine can work only on cytosine. These 4 chemicals modify these 4 bases. Once the modification has occurred, it's like kind of tagging that particular nucleotide. Say for example, DMS have methylated guanine residue, so it is kind of now tagged that this is the modified base. Now all 4 reactions would be followed by next chemical called piperidine, and piperidine has the ability to displace or break the phosphodiester bond wherever the base is modified. Okay, so if I add piperidine over here, now what will happen? It will find the modified base and it is going to cleave over here. It is going to remove the base from here. The degradation would occur and this fragment would go away. If this was my 5' to 3' strand and I have added dimethyl sulfate to it, there is a guanine over here that would be modified and followed by piperidine, and it will break or it will displace this modified base. So I would get only this much strand which I can read because I have labeled 5' and I'll not be able to detect this. And that's the whole purpose of labeling it. Again, this will become much more clear. As of now, you remember the mechanism that we have 4 different chemicals for 4 different nucleotides which modify the bases. followed by piperidine treatment. Piperidine can see wherever the base is modified and it will displace that modified base. It will break the phosphodiester bond and cleave that fragment. Okay, that's why it is chemical degradation or chemical cleavage method. Now, can we make this more clear with taking an example of a sequence? Yes, let's try and do that. Now, before we go ahead, take an example of this and understand exactly how these chemicals work. One important point over here is the chemicals that we are taking in each tube. That chemical concentration and condition of the tube is adjusted in such a way that it can act only on one nucleotide per strand. Okay, this is important. You will see why. So now, say for example, if this is the sequence that I'm working with, this is a single stranded form and radioactively labeled 5' end. In my tube A, I have added dimethyl sulfate followed by piperidine. So that means I'm looking for guanine residue in the strand. So suppose this is the first strand. Randomly, as I said, only one molecule per strand would be modified. So this DMS would find guanine and modify it, and followed by piperidine. Piperidine will see the modified base and break the phosphodiester bond and displace this base from here. As a result, you are going to get two fragments produced, isn't it? But this fragment, as there is no label, we are not going to detect it. We will be able to detect only this particular strand because that is where we have 5' label. That's why labeling was important. Alright, so this is the one fragment that we can get out of it. Second possibility is there is one more guanine in the strand. So this DMS can also act on this guanine and piperidine would displace this. So as a result, again we have this strand and this particular nucleotide also will be there, but will not be able to detect it. This is the friend that we are going to get. So at the end of each reaction in a tube, we are going to get different sizes of DNA fragments, isn't it? And this fragment size actually corresponds to the position of that particular base in the DNA sequence, isn't it? If the fragment is broken this much, I know that I'm working with DMS, that means it has modified guanine. So there was one guanine over here. Then there is another fragment which is longer than that. That means there is one more guanine over here. So the fragment size would actually give me the relative position of a nucleotide in my DNA. Okay, now how can we read this? That also we will see. This is one more example. In tube 2, say for example, we added formic acid followed by piperidine. Formic acid would modify both adenine and guanine. So wherever adenine is there and whenever guanine is there is going to get modified, and piperidine can displace this nucleotide. As a result, you are going to have different length fragments produced. Now you might be wondering how to know, whether it is adenine or guanine which was given by formic acid. That also we will see in a while. Now, still all this has happened in tubes only. We have all these things happening in reaction tube. Modification has occurred, cleavage has occurred, different sizes of fragments are produced. Say for example, in this, I got these many fragments. Here I got so many fragments. Here also I got different sizes of fragments. How can we now determine that where is this present in the sequence? So now what we'll need to do is again subject each tube to electrophoresis. Alright, so we have our reactions ready. We subject it to electrophoresis. What happens in electrophoresis? All the fragments based on their size would get separated. Shorter the fragment would move towards the bottom of a gel because it's smaller in size, it can travel more. Wherever the fragments are getting larger, it will be towards the top or near the well in the gel. So it is this much clear. Now let's say for example, once I process my sample, this is what I got as a result. Once I did the electrophoresis, this is what I got. Okay, these are the bands that are developed. How can I read this? Now look at this. We know that here we had treated in this tube with DMS So I know this well is for guanine bands. Here I had treated with formic acid. So I know here I'm going to find adenine and guanine. This is the lane where I loaded the sample treated with hydrazine. So I'm going to get cytosine and thymine. And this is the lane where I have loaded the sample which was treated only with hydrazine. So I'm going to get cytosine. But how to read it? As I said, the shortest fragment, as the fragment is short, you will find it at the bottom of the gel. We know this because the size is small. And when the size is small, we saw over here, it is closer to 5' end, right? As the size become big, it is moving towards the 3' end, right? So when you read the gel from bottom to top, we are actually reading the sequence from 5' to 3', because shorter the sequence, it is closer to 5' end, and as it becomes larger, that means the nucleotide is closer to 3' end. So we always read the result from bottom of a gel to top of a gel. That gives me sequence from 5' to 3' direction. Okay, this much is clear. Now how we will read this? First, let's understand how to differentiate that the bands that are present in these two lanes are of cytosine or thymine. Here it is clear, it is all guanine, it is all cytosine. But what about these two lanes? For that you need to compare. Say for example, we are reading for adenine and guanine, compare it with the guanine lane. If you find band in both the lanes, that means that is of guanine, right? Because that is the only common nucleotide in both the reaction mixture. Alright, so if you see band in both the lanes, that means that is of guanine. If you see band only in this adenine plus guanine and there is no band in this particular lane, that means it has to be of adenine, right? Because if it was guanine, it should have been present over here also. So similarly, if you see band in both these lanes, that means that is of cytosine, because that's the only common nucleotide. But if you see band only in this particular lane and there is no band over here, that means it is thymine. Okay, now let's try and read it from bottom to top. This is the bottommost nucleotide, right? There is nothing after this. So this is the bottommost nucleotide and the band is present in this lane, A plus G lane. So we will also check, is there a band in this G lane also? No, there is not. So that means this nucleotide has to be adenine. Let's write it over here. After this, these two bands are there. Okay, before this, these two bands are there. There are bands in both these lanes. That means it is of cytosine. Let's write it over here. Now look at this band. There is only one band in A plus G. There is no band in G. So that means it is adenine. Then you have band in both G and A plus G. That means it is guanine. Next is this in both the lane. That means it is guanine. Then we have only in C plus C. There is no band in cytosine lane. That means it is thymine. Then we have one band only in adenine plus guanine, nothing in the guanine lane. So it is adenine. Then we have band over here, C plus T and C both. That means it is cytosine. Then again, three bands I'm taking together. All these three are present in both the lanes. That means it is guanine, guanine, guanine. Next, in both the lane, that means it is cytosine. Again, both the lane, it is guanine. So this is the sequence from 5' to 3'. Let's write it over here. So this is the sequence of the strand that we are working with, 5' to 3' direction. So I hope this was easy to understand, right? Preparation of the DNA, where we labeled it, we separated it, denatured in single-stranded form, and then we isolated those single-stranded DNA, and then we divided them in 4 aliquots because we need to treat them with 4 different chemicals which are specific for each base. And in chemicals there are again 2 steps. One is modifying the base and then removing or cleaving at that particular modified base. By doing so, the fragments that are produced, that fragment size would give us the relative position of that particular base. So once we are ready with reaction mixture, again we will go for electrophoresis, and based on the size of the fragments the DNA would be separated. Then we'll go for autoradiograph. We will see the development of bands, and from bottom to top you read the bands. That will give us the sequence of DNA in 5' to 3' direction. It's very interesting, I think. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I'll see you next time. Until then, keep learning.